Thank you. Thank you all for coming back to this room. It's been a lot of fun. Thanks for the invitation. Yeah, so my lab in general, I like social behavior, not because I like one particular behavior, but as we're going to talk about, I really like all the different types of computation that exist in the processing and display of social behaviors, basically. Sort of any sort of computation that you can imagine in neuroscience, I think has to take place or impacts the animal's behavior in general, and particularly its behavior with others. And years ago, I really wanted to sort of understand how external sensory information is processed and how it moves through the brain and how the brain impacts the information to be able to make an appropriate behavioral choice. And that didn't seem like it was going to be too hard of a thing to do when I was a postdoc. But as you've heard this morning, it has been really hard. And my talk today is basically going to focus not so much on telling you the answers to how social behavior is coded, because I know very little at this point. It's going to be sort of telling you sort of the way that we're thinking about social behavior and the types of questions that we're thinking about, and how maybe we can offer a different voice to the field. And when I started this in Catherine's lab, we really did start with the behavior first. So I've always been staring at animals. And social behavior is a little tricky because you have two individuals, and their behavior is dynamic. So if I want to look in the brain of this guy, it really depends on some of the signals that this guy is sending. So if he's having a bad day, if his signals aren't so robust, if there's a lot of different variables, maybe this guy's brain activity is going to look different on day A from day B, and some of that could be some of the noise that we see in behavior, right? And so one of the early things that we tried to capitalize on was this ability of mice to be able to respond to the olfactory world with really distinct behaviors. And so there's a specialized subset of olfactory or smells that are especially salient to the mouse, and we call those just in general pheromones. That word in particular can be very loaded for some people, but just in general, what I mean is some sort of specialized scent that has a high probability of eliciting behavior. And mice are nocturnal, as y'all know, and so the olfactory system plays a really large role in their behavior. And over the years, we have isolated and either reduced to really highly purified fractions, or in some cases, been able to create synthetic molecules that are highly salient. So molecules for a pup to be able to recognize the mom and initiate suckling, molecules that promote aggression, molecules that are emitted from other species and promote, in this case, I would say it's probably the only innate behavioral response that I can be certain of. Our mice are all lab mice that have been raised since the 1930s, in a lab environment, so hundreds of generations. And if you expose them to different predator odors, they respond very strongly with very strong behavioral signals. So that I would call true innate behavior. And then other labs have identified different chemosignals that promote mating and different affiliative behaviors. And so all in all in the field, even though there's probably hundreds of different olfactory cues that the mouse is capable of detecting that are probably promoting behavior, our field has about a dozen isolated cues that sort of cross these different lines between within species, across species, positively valenced, negatively valenced, and so this repertoire is sort of our toolkit to be able to replace that second noisy partner and initiate a high probability behavior with just an olfactory cue. And the goal is to be able to scale it up, scale it down, present the same cue over and over again to animals and get them to do a behavior. Now, as Dayu said earlier this morning, certain behaviors we cannot reduce to just an olfactory cue. So olfaction is necessary for aggressive behavior. If you impair the olfactory system. Even with all the visual cues, it's very difficult or near impossible, as far as I know, to get a mouse to attack another mouse. But the olfactory cue on a cotton ball is not sufficient. So it does need, there's some sort of context, some sort of sensory cue, and some sort of movement is generally necessary. So these cues are important, but they're not everything. Okay, and the idea was that we would take this, this now sort of purified stimuli in the environment, the simplified stimuli in the environment, and we would walk forwards and find the circuit, and within the circuit, what we imagined is that there would be nodes that would be dedicated, perhaps at the periphery, to informing what's out there in the sensory environment. What's the chemical cue and how much of it is there? So, and then before the animal should decide on what behavior to perform, we have reasoned that it needs to make a bunch of different decisions. So it needs to consult its other sensory systems and make sure that it's not detecting an illusion. If you can smell a cup of coffee, can you see a cup of coffee? Can you touch the cup of coffee? So is there coherence with the other sensory systems? The animal needs to decide if it's experienced the cue before. Does it have a memory of it? And was that experience good or bad, positive or negative valence? It also needs to sample both the olfactory systems and the other sensory systems to make sure it's the most important thing out there. So if it smells a mate, but if it smells a predator, it needs to decide which to act on. And it needs to consult its internal state. If it's dominant or subordinate, pregnant, stressed, hungry, it's going to respond to these cues differently, and that needs to be factored in. We can imagine that there's a bunch of other decisions that the mouse has to make, we'd like to brainstorm and think of, if we could exhaustively list those decisions, but those are the easy ones that come to mind. And then at some point, you can imagine all that information is integrated and that some sort of command is made and the animal's going to respond in some way, either with aggression or mating or do nothing, depending on the environment. And so that's the general strategy. And that sort of processing of what is each node good for and how is it achieving its goal is sort of what my lab would like to address. One of the things about pheromones that I didn't appreciate when I first started working forever ago was that I'd sort of imagined, I think the field had sort of imagined that the response to them would be sort of robotic. The animal would smell the cue and a flip would be switched and the behavior would be, a guaranteed, right? So in the back of the magazine, you can look for, there's ads for male cologne. If you wear this cologne, it's pheromone cologne, females are gonna just fall all over you, right? That's sort of, how you imagine they would work in mice as well, based on those sorts of ridiculous claims, right? But it's not the case. And you heard a little bit of that this morning in the first two talks. And so I just want to emphasize too, that there's a lot of factors. So these odor cues have a very high probability of initiating a predictable behavior, but only in the correct context, really. And so for aggression, male-male aggression, the animals have to be a certain age. Juveniles, you can give them all the odor cues and give them another moving object, and it's very difficult for these odor cues to initiate any sort of rage at all. Gender, females don't respond to the cues the same way males do. And experience, dominant animals respond differently. Whether they've mated, they respond differently. As Dayu said, whether you singly house them, they respond differently, right? So all these factors feed in. Mating is the same way, that's pretty obvious. And even parental behavior, these are all biased by certain things. In the case of male parental behavior, experience really biases the ability to perform that behavior. And so, so this can be a flaw if you choose to look at it that way, right? That you have to do the behavior just right and it only occurs in this one context. Or it could be a feature, right? Because what you have are that all of these different modulations are feeding in onto the circuit someplace. And how these things are taken into account and coded for, how they're changing that probability of behavior, we consider to be really interesting and add to the richness of the system. And so all in all, what I want to stress here is that behavior, even in the mouse, is kind of personalized based on whether the animal is stressed or hungry or dominant or subordinate. And the types of things that really feed into this sort of personalized response are, as I mentioned, experience, the developmental state, and that internal state. And so over the past more recent future, my lab has really chosen to focus on trying to determine how internal state is affecting or modulating behavior. And so, you know, if you sort of take this ridiculous schematic here, you know, the question, you can reduce the question down to where's your internal state represented and how is it represented, right? For something like hunger state, maybe you'd look for certain hormone receptors. For something like experience, such as dominance or subordinates, you know, maybe that's using a whole different type of mechanism, and you'd look for that in a different place or in a different way, right? Okay. So to start this, we messed with a bunch of sort of canonical changes in internal state. And the postdoc that was working with me at the time, Sandeepa Day, decided that she really wanted to focus on female behavior, so the type of behavior that Susanna talked about this morning. And that's because there's a really nice control and really rapid change, as she showed you, of internal state. And so what we knew is that male cues attract females, but they don't always attract females, right? And as she told you, that's dependent on their change in the ovulation cycle, the estrus cycle in the mouse. And as she mentioned, in our lab strains, this is a really fast cycle. So to go from estrus and go back into estrus, it's generally about 5 days in our lab mice. And so every couple of days, the animal is in the opposing stage, which we call diestrus. And so she showed you the receptivity of the animal to males, and we can reduce this down to odor cues as well, simplify the environment. And so if we take a complex blend of male odor cues and we place it on one side of the arena and a control odor on the other side, you can see that when the animal is in estrus, so when she's in the receptive stage, this is a trace of where she's moved, and the warmer colors indicate where she's been the longest, and you can see she's loitering sort of around these cues. And just a couple days later, this female is sort of indifferent to these cues. She's not loitering around them as much, and that's when she's in diestrus.
So how do you recognize these different states?
The female does it for us. So she's just naturally cycling in and out of this ovulation cycle. So this is her hormonal cycle that's regulating ovulation. And so when she's in estrus, she has a surge of hormones, which I'll tell you about in a minute. And then just a few days later, naturally, without doing anything to her, without any training, you know, just by her internal state changes, the same individual is responding to the environment differently. And that's what we were interested in.
I think the question was, how do you know which stage she's in right now?
Sorry. There's a lot of, you can get down to it precisely by measuring female hormones in the blood system. We don't do that. We just do a vaginal lavage, and the cells that you get from doing that procedure look different in each of the different states. It's very easy. It's the females don't care, and it's very rapid. Yeah.
Maybe it's a very naive question, but can you spend a few words defining internal state and how you identify an internal state? Like, you use behavior to assume an internal state of the animal, or markers to claim that the animal is some issues?
Yeah. So I cannot spend a few minutes defining internal state. We could stay here. I could literally go on like a 5-hour rant of what we know and what we don't know and how complicated it is right now. And I think that's one of the questions that I have a student in my lab that is trying to ask that very question: what is an internal state in different ways? But for this, I'm going to define it as these physiological changes. We can measure them by hormone, as well as these behavioral changes, and that those two things together are good enough for me to just generally use the word. And we have a very tight definition of how those cells look like when we do the lavage, so that it's in one state or another. So we can get animals that have very, very similar behavior. Mm-hmm.
Can I just ask you one thing? Because sometimes people distinguish between behavioral estrus and physiological estrus. So I know I'm being picky now, I'm asking, but like in a situation like this, So how would you like turn around this question? Like because sometimes the females like the lavage can tell you something, but then their behavior is not she's not receptive, and this happens several times. So like again, I know this is it's a 5-hour or more discussion, but.
This one's easier for me. So for this, with the odor cues, pretty much all the females in that certain, when the cells look a certain ways that we consider to be estrus, they all respond with a general level of attraction. So we are not, in any of the stuff I'm going to tell you today, we're not looking at the female's behavior with the male, just with these odor cues. And the only behavior that she does is just attraction or sort of this map behavior, And so those really more complex behaviors that you're looking at and that would have other things that impinge upon them and maybe have to change our definition of the state, we're not even looking at. We're totally superficial here. Okay, so what we found over the years is that within this complex odor blend, there is a subset of cues that are attractive, and the female brain is gating on those cues during this estrus state. I'm just going to keep calling, I'll just call it the estrus state, but we now know that there's some limitations with that. And so she's gating on those attractive cues at this time, and she's not attending to them in this time. Okay. And so super, this is exactly what we want. So what we have is we have this one individual, and there's something being modified in her brain or changed in that individual's brain that's rapid and that's changing her response to the environment, the sensory environment. And so we had our system, and I was all set to ask, where in the brain is this occurring? Awesome. But my postdoc kept hounding me and telling me that she really wanted to do this one control, and she wanted to make sure that there were no changes in the sensory system before she started looking in the brain. And we had these long discussions about how, I really appreciate controls, and formally that is a great idea, but in fact, it'd be silly to assume that there should be any sort of modulation on the sensory system, because the function of a sensory system, I apologize, is to basically vacuum up as much information as you can about the external world. The more you get, the more the brain can use to make a good choice about which behavior to do. And so that assumption that I made has been based on lots of different kinds of human experience, and our understanding, an illustration of this comes from the cocktail party phenomenon. And so here's an example where you're in a really crowded room and you're having a conversation with someone in front of you, but everybody else is also having conversations. And so what happens is you're able to selectively attend to that person in front of you by sort of paying attention to them in such a way that the rest of the conversations sort of recede into a din. Yeah? I don't. Hopefully you've experienced that. But if somebody across the room says your name, you can hear it. You can know what direction it came from. You may know whether it was male or female that said it. If you know the individuals there, you may even know who said it, right? And so that means that you, even though all the information receded to a din and you were able to focus on this aspect of the sensory environment, your brain was getting a lot more and it was being gated out, right? So there was an attention gate of some sort. And so that's what we expected for the olfactory system. That was the model we had, is that the sensory system in the periphery is good for data gathering, and the brain's function is decision making. Okay.
But I think that's just a hypothesis, right? It could be that states also directly adjust and modulate primary sensory organs. I mean, I agree with you. It's sort of less. Doesn't make as much sense in terms of information processing, but you can't exclude it.
Exactly. And it's never a good idea to interfere with the postdoc's desire to do an experiment, right? And the experiment was really easy. So and she didn't need my permission to do the experiment. It's just that, we had these discussions beforehand going, time is precious. But the experiment was really easy because the nose, we had been messing with the sensory neurons on the nose. They're easy for us to just quickly screen and make sure that there were no differences between their activity during estrus and diestrus. So the difference between, one of the major differences, although there could be many, and there are many, between estrus and diestrus is the production of the female hormones that are occurring from the ovaries. And during estrus, you get the surge of estrogen. During diestrus, you get a surge of progesterone, shown here in black. And so we could, she removed the female hormones so that there was no bath of hormonal information whatsoever, and then by overreactomizing the animal. And then we were able to just do an ex vivo prep and calcium image the neurons from the sensory epithelium. So this is from the nose, a specialized organ in the nose that we knew detected these male attraction pheromones. And so this is rapid, these aren't cultured. And so these cells came from a mouse that had no hormones, and she just did a 1980s experiment where in the bath she could add back hormones, and we could say, you know, is there any effect of hormones, and what happens when there's no hormones? And this experiment I love, so I'm going to show you the raw data for this. So these are calcium imaging traces of individual neurons as we perfuse this attractive cue in the dish. And you can see that here this subset is responding, they're responding. Then she adds the progesterone. She waits just a little bit of time, 15 minutes. And then she perfuses again with the cue, and they don't respond. We made sure that they just didn't die during that time by adding this little ligand that can directly gate the channel, and sure enough, they're all alive and they can respond. And so the reason I love this experiment is, remember, this is ex vivo, so these neurons are in a dish removed from the brain. So the brain is not telling these cells that there is progesterone around or that she's in diestrus. These cells must have detected this progesterone all on their own. So it's acting directly on these sensory neurons, which was surprising to us. And then second of all, it's happening really rapidly. So most steroid hormones act through transcriptional regulation. They can take an hour to happen, and this could happen in literally as little as 5 minutes, but it was pretty much a rapid action signaling. And so we were very curious about the mechanism for that. And so over a great body of work, this is what she found. So this is the signal transduction pathway on these sensory neurons. Sorry to give you molecular biology. We'll get to more system stuff later. So that's a G protein coupled receptor that's specific to detect the attractive cue that's out there. When it detects the active cue, it goes through the signaling cascade, through G proteins, phospholipase C, and gates that channel. Okay. And what she found is that in addition to that stuff, there's another hormone receptor, a non-canonical hormone receptor on these sensory neurons that's detecting progesterone. And when it binds the progesterone, it starts a kinase cascade, and that phosphorylation ends up phosphorylating the PLC as a target. Okay, so the first state, when there was no hormone around, is what's happening here and here. Then she adds the progesterone, it binds, it does its signaling, it phosphorylates this and turns it off, and that's what's happening here. So no matter how much Q is in the environment, the female, she doesn't know because her channel hasn't been gated and her brain has no idea. So she's just blind to it. Okay, so this is a summary of what we found. During estrus, she can smell these attractive cues, her brain can sense them, and she does the attractive behavior. And during diestrus, her sensory neurons block the ability for her to detect this attractive cue, and that simple gating right at the periphery doesn't give the brain any information, so the brain doesn't act on it, and she's pretty much indifferent to the environment. Okay, so this was stuff that we had done years ago, and this really bothered me. So, we took this all the way down to a phosphorylation event because I just didn't understand why a sensory system should deprive, even though it's possible, why should it deprive the brain of information? Why would you evolve the expression of this hormone receptor in these cells and turn off this system? It leaves the female super vulnerable, and it doesn't allow for any alternative decision making of being able to sample the cues and act, independently, right? And so I've gotten a lot of answers over why this should be. Most of them is that these poor females probably would be distracted by the male information. And so if they were to sense the male during diestrus, they would, maybe their brain would be so preoccupied with the thoughts of the male that she just couldn't do her daily stuff, and it just wouldn't work out for her, right? She'd die or something, I don't know, right? And so, I think that that's a little ridiculous, because think of all the sensory information you're getting. We use the term sensory overload, but it really takes a lot, or even is that a thing? I don't know. There's a lot of information that you don't attend to, or that it doesn't necessarily hurt you to have it. In fact, I would say that, again, back to my original premise, the more sensory information that you could take up, in channels that we don't even have. If I could see in UV, I would be delighted, right? The more I could get, the more you can act on it. Okay, so I didn't think that was reasonable. The second thing is that maybe there's something about detecting a male when she is in diestrus that would make her unfit. And there is this physiological phenomenon called the Bruce effect, that once a female has mated with a stud male, if he goes off and a new strange male comes in within a certain period of time, she will spontaneously abort the fetus. His male pheromones cause her to go into estrus, and so she will lose the pregnancy. And that's thought to keep her fit, because if she ends up putting the effort into siring all these pups, the 3 weeks of gestation and the metabolic costs, he can recognize that they're not his, and he would just eat them kill them anyhow. And so that's what's thought about this physiology. And maybe there are other aspects of physiology that we don't fully understand between estrus and diestrus and sensing these cues that would be a cost on her fitness. And that's all possible, but it seems like there's other ways to engineer this, especially deep in the brain, rather than going out at the sensory system, right? And so, so for several years, we've been sort of focused on, maybe this thing that we saw specifically in the female behavior, maybe that's not just specific to female behavior. Maybe this is something that this olfactory system does. And so for the last year, we've been considering this, and I'm going to show you some half-baked data that isn't quite ready for prime time, but it's starting to make us feel that maybe we could be onto something. Okay, so one of the first things that gives us confidence that something larger may be going on is that the VNO system looks a little different than other sensory systems. So these basic canonical sensory systems that use sort of these large families of G protein coupled receptors to be able to sense the environment. They sense different sensory ligands by an expansion of these GPCR repertoires. So in taste, there's 60 different GPCRs. In the main olfactory system that's being used to sense everyday associative odors, there's about 1,000 of these in the mouse. In our pheromone system, there's about 350 in the mouse. But they're all thought to couple onto this module of signaling that is, no matter what the receptor is, this module is unchanged. Yeah, and so in the vomeral nasal system, it's thought to, as I showed you, signal through these G proteins and PLC beta 2 and onto that channel. And so we looked at that a little bit more carefully. And from our initial data, if that's the case, if there's just one of these PLCs, and all of these sensory receptors are funneling onto it, and the female system is shutting it down during a portion of her everyday cycle, right? Then she would be vulnerable. This system is important for detecting predators and for detecting other types of very important social information. And that means she would just be blind to this social information for a portion of her everyday experience. And that just seems a little ridiculous, not ridiculous, but a little sad, because I really kind of wanted to fight for these little females and hope that they were getting the same cues as the males at all times and able to be fit, right? And that turns out not to be the case. So in this system, if we manipulate that PLC beta 2 that we did show was important for the response to this attractive cue. So in animals that don't have PLC beta 2 just in their nose, we find that they are unable to detect and respond to it. So all of the neurons that sense the attractive cue are using this PLC beta 2. But if we use other stimuli, here's complex male urine, and in it, half of the signaling's gone away, and that's through this attractive cue and some similar members, similar ligands that are in the family of this attractive cue. They disappear, but there's still a bunch of neurons that are detecting and responding to cues. They're not silenced. They're not being phosphorylated and inactivated. And then if we look at predator cues, thank goodness she can detect and respond to them just like a wild type animal if she's missing these cues. So this is telling us that PLC, that not all the receptors are funneling into this one module of signaling. And it turns out, our field was a little ignorant, there are 16 different PLC variants in the mouse genome. And beta 2, the one that the field had assumed was important for detecting pheromones, is this little guy here. And if we just grind up the total VNO and do RNA-seq on it, we find that almost all of the PLCs, we find reads on them. And there's quite a few that are much more highly enriched than that one, right? So PLC beta 2 may be the one in this attractive cue sensing neurons, and this one may be the one that's the module for all the rest. But if we took a little more resolution, instead of just looking at the total organ, if we sequenced single cells, what we found was that we still saw this whole repertoire of PLCs. So there isn't just beta 2 and then a module. There isn't a single module. There's a lot of these different ones. And it turns out that there's largely one per cell. So here's a heat map of the number of the reads that we see. And what we find is that there's some cells that are expressing 2, but most of them are expressing really just one of these genes that are coding PLCs. Okay. And we could confirm this by doing an RNA scope experiment. You don't really need to see this.
There were a bunch of cells who didn't express any, is that right?
Yeah. So this is weird in that we see about 30% of the cells that aren't expressing anything, and that could just be, these are expressed at a very low level, and a null read response could just be, a function of the sequencing. But we kind of don't think so. We think there's something else going on in a proportion of these neurons.
So you think they have a different one that you're not looking for? Or
I think they have a different kind of signaling altogether. So here we're showing that there's a variety of ways it can signal, and then there is this subset that looks like maybe something else is occurring entirely differently, but we're not actively.
Have you verified that these cells actually respond to calcium imaging?
So no, these are if we calcium image before we before we transcriptionally profile, the quality of the RNA goes down. So we have done cells that way, but we haven't done a lot. So we have tested cells and then sequenced them, but these have not been activated at all. But these are all selected for cells that are transcriptionally healthy and look like mature neurons. So they have a single receptor, they have all the other signaling elements that make them a mature neuron.
But you don't know whether they actually have ever responded.
We don't know whether these guys have ever responded. Yep, we don't know. So we could confirm this in a different way just by doing in situ here. This is RNA scope, and these are at very low reads, so the responses are punctate. But if I go ahead and sort of do a cartoon diagram of what we saw was very much like what we saw in the sequencing, in that we pretty much get one per neuron. There's an example here of where we got 2 PLCs. We also see them at different levels, so there's some PLCs that are expressed in a lot of neurons and others that are expressed in just a few neurons. And so by these 2 methods, what we think is that this is different than the other systems in that we have a lot of different receptors and then we have a lot of different signaling molecules. Okay, so why this whole family of signaling molecules? What does it matter whether you have B2 or E2 or L1? And it turns out that the PLCs are really divergent from each other. So these are separate genes. These aren't like a single gene variant or splicing variants. And at the sequence level, they're very different from each other. It's very easy to make probes for them. And they can be clustered, so not all of them have been studied well, but what we can find from the literature is that they can be clustered to those that are largely signaling through G proteins and others that are even responding to entirely different upstream molecules, so receptor tyrosine kinases. The ones that are studied that we can find have been known to all to be modulated by phosphorylation, just like we saw that silencing by phosphorylation on beta-2. But depending on their residue, they can be not just silenced like we saw before, but they can also be activated so that without phosphorylation, their basal state is off, and with signaling, they get turned on. And in some of these variants, it depends on which residue they get phosphorylated on, and so they can go bidirectionally. And so this gives us this idea that there's this whole family to give this flexibility in the system so that some neurons, like the ones that detected the attractive cue that we saw, they could be silenced when progesterone's out there, but maybe there's other cues that are responding to different things and they could be activated. And so for us, that gives us sort of a rationale of why there would be a bunch of these. Okay. The other thing is that we looked at their spatial distribution because the spatial organization of this organ here, so it's in this sort of crescent moon shape here. These are probes that recognize a bunch of different receptors, and so you can see that these probes only recognize families of the different molecules, but there's about 350 different types of G proteins within this system, and they can be grouped largely into two families that are recognized by these different probes here. And the important thing about this is that these two families project their axons into the first relay in the brain to different places. So they're right next to each other. This is all the accessory olfactory bulb, and the green ones here are going to one region, and the red ones here are going to another region. And we think that these are functional differences in their output and the types of behaviors and the types of ligands that they're coding for. And so we could look at the spatial representation of these PLCs, and we can find that across this crescent moon, we find that they do have restricted spatial distribution. So here's a nice clear example of this one here, PLCL1, that is restricted to this apical region here, and that would overlap with only one of the receptor families. So it would only be able to, it looks like it's not stochastically or randomly chosen to be expressed with any receptor. It's restricted to be chosen with a certain subset of receptors. And here in this green one, it's in that opposing larger level. And we are currently using different techniques to refine this rather into from just that big green stripe and red stripe to different individual receptors and see how determined the two are. And we expect that if we see some sort of logical organization between receptors and PLCs, that would give us greater confidence that they're there for a purpose, a functional reason. Okay, finally, the third player that we looked at that makes us think that the system may be bigger than just female hormone detection is the receptors themselves, right? So here is this non-canonical progesterone receptor. It's expressed in a lot of the neurons, but when we did our RNA-seq, what we found is that there are a bunch of other really interesting hormone receptors that are expressed in subsets of these neurons. And so we see receptors of neurons that would be able to detect the stress level state of the individual. So the whole suite of adrenergic receptors and corticosterone receptors are expressed on these primary sensory neurons in nice levels, as well as a whole suite of receptors that can detect the hunger satiety state and act on that, as well as other interesting families of receptors. So we see more than just progesterone. The system has the molecular capability to be able to detect the internal state across a lot of different axes. And so what I've shown you is that the system is weird in that it's using a lot of different downstream signaling mechanisms. We think that there's at least some level of genetic determinism or predetermined coding between the identity of the receptor, which is going to tell what sort of cue the cell can respond to and what sort of PLC it's going to represent, and that there's all these wonderful richness of hormone receptors that could modulate the system in response to a lot of different behavioral states. And so the real question that we would like to get at at with this is, is this a global system to modulate perception, like the modulation of perception with that male attraction cue. And so, again, this data is super preliminary, but this is the model that we had, and we decided just to test this completely grossly. So for this, I'm really taking liberties with the use of state here, and we decided that we weren't really sure which axis to push this in if we wanted to look for a global system. So we just grossly said that juveniles should have one sort of hormonal milieu, males, estrus females, diestrus females, and pregnant females, all of these guys should have different hormonal balances, yeah? And if you accept the model that signaling should be sensory signaling should be a big vacuum, and that you should take in whatever cues are out there, everybody should have the same percept of the environment, what you would imagine was that if you looked at the activity in the olfactory bulb, where this information, at the level of the first relay, that the representation of all of these different animals to a certain chemical cue, if you gave them all the same pheromone mix, the representation would be the same. You would see the same patterns. And so we did this with help from Julian Meeks at UT Southwestern. And this is just expressing GCAMP in the sensory neurons. And these sensory neurons in the olfactory system, you saw with that colorful sort of crescent moon, they're distributed. But all the receptors of the same type converge at the first relay in the brain in a neuropil. So all the receptors that would spell that attraction cue go to one region, and what these dots are puffing of odorants, different odorants, so you're getting different patterns, and these dots at a most minimal level represent a single olfactory receptor type, so we call this glomerular. Okay, so if we did this, yes?
Was it ex vivo?
This is ex vivo here, yeah. And we have begun playing with mini scopes, and right now our angle is totally wrong and they're way too heavy because the olfactory bulb's really at the front of, and the animals just keep falling over. It's the saddest thing ever. So we'd like to, this ex vivo thing doesn't allow us to play with the hormonal state, and so this was just sort of a one-off preliminary. Okay, so here's a snapshot of what we saw. So across this y-axis are different blends of different combinations of things that we know activate pheromone sensory neurons. And what you can see, if you gauge just right here, you can see there's a lot of noise in this whole system. Down here, this gets really blown out, so the whole system, this is a little noisy still. But what we see is that to this one set of, these are three synthetic ligands, pure ligands, that activate the sensory neurons. And in juvenile males, you're getting this robust representation. They're detecting, there's a lot of receptors that are detecting this cue, and they're getting this broad pattern, whereas these pregnant females are hardly detecting this blend at all. Okay. So this is just preliminary data that says that we have this system where there's a little bit more to it than just the canonical signal transduction pathway. We've got 3 different places where you get variability, and we think that the variability in the system is allowing for modulation of perception.
How old are your juvenile mice? So
3 weeks.
3 weeks. So they are not sexually mature at this age. Okay.
Yeah, they were exactly 21 days.
So in RNA-seq data, you can tell like which receptor and which PLC and which hormone receptors
Yes.
they are correlated.
Yes.
Right? So can you find certain patterns like
Yes.
this receptor is for predator detection, and then there is this stress hormone receptors are being co-expressed with it, and then there's this PLC associated with it.
So when we started this project a year ago, this was going to be a fast, easy, and safe project because all we had to do was a single-cell sequencing experiment. And we thought that if we could, we would be able to get the organization of those 3 things, and we would be done. But we have, there's about 50 different hormone receptors that we think are interesting that are expressed, as far as we can tell, in sort of these salt and pepper all or none. They're either expressed in a cell or not. So that's our criteria for whether it's just a general hormone receptor or something that could be interesting here. Then we have the 16 different PLCs, and we have 350 different pheromone receptors in the system. And so the calculations, once we start, so our first couple of sequencing attempts with 10x, 10x is right at the level, if we get 1,000 cells with 10x, it's right at the level of being confident that we have enough PLC reads, because those are at very low read levels. So we have to, we can't go above, several hundreds of cells even with 10x. And so the math we did to be able to get enough replicates of each receptor type was above 75,000 neurons was our sort of back of the envelope calculation. And we needed to deep sequence those, and I'm poor. So, shallow C, 10x, we could do it, but deep sequencing of 75,000 cells, it just wasn't going to work. And so we have genetically labeled cells that we just haven't sequenced yet, but we've got some other tricks now to try and take this fast. But that was the easy right experiment to do. We just the numbers just were, I just couldn't pull the trigger on those numbers. Okay, so here's what I do not know is happening, but I think might be happening, and what we're looking at. And that is that the system of the way we were thinking about it, the sort of passive sensation, and it is sort of, what you sense alters how you feel. So you smell holiday smells and you're a kid and you get all excited. You smell holiday smells and you're adult and your in-laws are coming to visit and you still haven't bought all your presents and you get stressed, right? Like that is the smell comes first and then that affects behavior. And what we're thinking is that the behavioral state is altering what you smell. So how you feel alters the percept, and then that's going to lead to different behavioral actions, as it did with this female sensing the male attraction cue. So we are still working on that. I don't know, it could be that all of these other hormone receptors and these PLCs are just there for decoration, and so I think it's going to take us about a year more to make sure that they're functional and actually doing something. Yeah.
What do you think about competing orders? Like these two hypotheses say like what you sense alter how you feel, and then you think about fear, a fear odor like cat smell or whatever, and how you feel like if you smell at the same time a cat and another male, how do these things compete? Maybe like shutting down reception of male odors can help discriminating other odors that in this specific time period can be more relevant for the animal.
Yeah, so you have a lot of really interesting questions in that sort of one bigger question. One would be the hierarchy of behaviors, right? So and those are sort of the questions that we wanted to look at the circuit level. We can imagine that a lot of that could take place at the circuit level, but and none of the stuff I've shown you here would suggest that that sort of hierarchy is occurring here. And then the second thing is that, because I say that this may occur, it doesn't mean, I'm not saying that this can't occur, so that if you smell a cat, you still cannot be afraid. What I'm saying is that the information about the environment may not be equal to all individuals. And so those differences that you see in the resulting behavior may be simply to this phenomenon that we hadn't appreciated before, the sensory gating, so that they're each getting a different representation of it, and that's why they're acting differently. Right? Yeah.
So, have you done any experiments in the system? Because if I think about the pregnant female, and just from my own experience, I can say that you all of a sudden smell much better when you're pregnant. So I'm wondering if you have done anything on how this is developed in the female, that basically the pregnant state is doing plasticity to the system, and like regarding your hypothesis that it doesn't mean what you smell but how good you smell, that they may be better in an odor discrimination test or something like this?
Yeah, so there's a lot of psychophysics in humans, and there are odor discrimination following different state manipulations, and all of these things are thought to be occurring centrally. And it's possible with some of this that some of these differences, there could be a mechanistic basis for some of these differences to be occurring peripherally. So none of this controls that we did where we removed the progesterone receptor so that the female was always attractive even to the cues, even in diestrus, she still wasn't receptive to them behaviorally. So all the things that Susanna talked about, all these changes, these state-dependent changes on the brain, they're still occurring. This is just sort of another level on top of it. And so the way that you described your personal experiences with a change of olfactory percept based on whether you're pregnant or not, you hear that a lot. We don't have the specialized system. Humans, so almost all terrestrial vertebrates have some level of it, whether a little one or in rodents, it's very highly evolved. This second one, rodents also have a second system that we have that's called the main olfactory system, and that's what they're sensing regular smells, not these specialized odorants, but things like peanut butter and things like that. And so when we look in the mouse, in the main olfactory system, we also see a very large repertoire of hormone receptors. Some of them are different, some of them are overlapping. And when we look at human neurons, we also see these hormone receptors. So our behavioral repertoire and our understanding of the cues is pretty well understood in the specialized system. We're going to look at that first, and then see if there could be some sort of modulation in the main olfactory system. So, so I don't know whether, I would say that those sorts of modulations that humans experience are probably happening centrally. I mean, it would be horrible to think that we're all getting a different subset of sensory cues, that our brain is missing out on these sensory cues, right? Because what's the brain good for if it's not decision making? And so, so why not evolve this out and let the brain have flexibility, right? Because this prevents any sort of behavioral flexibility. If you gate out those cues, the brain can't all of a sudden respond differently.
So kind of related to that, do you think the difference between what you're describing here and like the cocktail party effect that you were explaining at the beginning of the talk might be the different kind of specific behavioral relevance of these different sensory systems? But in the pheromone system, you have specific smells linked to specific behavior, whereas in the auditory system, in most cases, it's not like that, although it might be, for some animals.
Yeah, I mean, in the other sensory systems, you don't want to be gating out information. If you in other sensory systems, right? Like, you just gorge yourself on hamburgers at a buffet. There's lots of different choices. You choose the hamburger, you eat a lot of them, you're satiated, and now all of a sudden you can see the rest of the buffet, but you can't see hamburgers, right? Like, that's sort of what's happening here. That's ridiculous. Your response to them may change, but you can still perceive them. This they're not perceiving.
I know, but I think there are examples in other sensory systems in animals where information is gated at the primary sensory level. And for example, in somatosensory gating during swims, during motion, is where the information you sense is a direct consequence of your own action. And then it often makes sense to just gate it out as early as possible. This information is relevant.
But a lot of that's thought to be gated. It's not thought to be gated in the primary sensory neurons.
But it could be.
It could be.
No, it would make sense. In that case, it would actually. So I mean, there are different ways where you can gate sensory information at that level. And I think in some instances it is at the very early level, right? Not at the primary neurons, but I think in the inner ear, right? The hair cells in mammals can be influenced, gated, and tuned by descending by efferent pathways. Yeah,
but that's different. That's feedback from the brain. So the brain is making the decision and feeding back.
Feedback from the brain.
This is not feedback from the brain. This is happening cell autonomously by the receptors on the sensory neurons. That's the weird difference, right? So that feedback from the brain, that occurs widely. So the brain's already gotten the information and it's acted on it, right? I don't want it anymore.
This might be a bit naive, but what we do know from psychological studies is that if the brain keeps on making decisions, at some point you reach this level of decision fatigue where you just don't care anymore. And I think in this context it might make sense to gate out information before the brain has to deal with it. Is that something that you also observe in rodents?
Well, no, I mean, I don't have an example of decision fatigue, or I mean, I don't know a paradigm for that. But you know what what we've been thinking about is that maybe this is an explanation for what this specialized sensory system is good for, right? You have this main system that actually does detect quite a lot of socially relevant sensory cues. In fact, the main system is probably the primary detector. And the VNO detects a subset of those cues and influences behavior differently than that behavior that's going through the main system. And we'd like to think of it as maybe putting an attention stamp on it, and that maybe the female isn't totally blind, but she's getting this attention stamp through this modulated system, and that leads to differences in behavior, right? And so that's sort of how I reconcile my tension with trying to figure out why evolution wouldn't let the brain do its job, and you know, maybe it is really happening. I mean, I love the fact that you guys are all like, this makes total sense, but to me it's still, like, it just seems like a waste of the brain.
So, just to follow up on Florian's question, so obviously, as you know, pheromones are absolutes. They're blends of things, they exist in balances between the two different sexes, and things are balanced out in a state-dependent manner. They don't kind of, they're not dotted in or out in a digital manner. And so I'm wondering, I've always wondered, how animals gate out self-cues. You know, I'm a source of robust odorants, and I'm a smell myself. Especially if you're a female, you're generating behaviorally relevant VNO detectable cues all the time. Do you think you've kind of, part of what's going on here is a specific gating so that you don't smell yourself and therefore behave in some ways as if you're smelling a female conspecific, for example, like if you're a female?
Yeah, no, we think for that problem, it's just habituation. So there's sensory habituation. And so, you know, if you walk into a room and there's baking cookies, you smell them for a little bit, but after a while, you don't really smell them so much, and your system is just habituated to it through molecular ways and through, in the sensory neurons themselves, as well as with some feedback from the... And the vomonasal system habituates as well. So we think that that's the mechanism for that. We think that this thing is responding to these hormonal fluctuations, and that's what that's doing.
So you're saying this does not come from the brain, it comes, but isn't it, if it's a self-generated hormone, isn't it
it's in circulation.
Huh?
It's in circulation. So this is highly vascularized. It's got mucus around it. So the ligand is there. Any sort of ligand that's floating around the body can get to the VNO.
No, but isn't it ultimately also generated by the brain, the progesterone, or whatever it is that gates these receptors?
But it's the circulating progesterone from the ovaries that has the major effect. So if we remove the ovaries, then the effect changes. Yeah.
So I'm just wondering, you said it's not the brain that tells them to shut down, but it's the ovaries. Is that your point?
It's the signals from the hormones. Yeah, it's the signal from the ovaries, the circulating hormones.
And that has no crosstalk to the brain. It's not. So
the ovaries are also talking to the brain.
But the brain talk to the ovaries.
And the brain is talking to the ovaries.
It's an indirect signal from the brain, right?
Yeah.
It's not, okay.
The brain can still be in charge, but the modulation we can get it to occur in a dish without the rest of the brain. And the receptors that are doing this modulation, the signal transduction, are right there on these sensory neurons. And so this modulation, this silencing, and potentially this activation through the other guys, that's happening right there. Without, and that's we would expect that the system would be modulated, at different nodes or maybe from feedback, but to have it right there at the sensory system so that the brain is really blind when the neurons are silenced. It's blind. Okay, so the reason that I drug you through that whole molecular tale of excruciating physiology. And not the interesting experiments that you want to hear about the brain, is because I think this sort of, that simple view that I presented, which we all knew was a simple view, but was somewhat reasonable about sort of, information flow being sort of progressive, feed forward, maybe linear, maybe starting from sensory information and working towards higher order decision making and integration, that may not be the case in this system, right? Because what we're getting here is really if-then decision making. If progesterone, then don't respond. If what, right? Or possibly more. Decision making that is occurring right here in the nose, outside of the brain, right? If that computation or that decision can occur here, all bets are off for where everything else is occurring, right? Things could be happening at multiple places and can happen in unpredictable order, right? And so, and the other useful thing about this is that if we do find that the system is able to be dynamically responsive to a bunch of different internal states, if we know about it, then we can that could be leading to some of the noise within the system and really impairing our ability to be able to understand it. If you grab two individuals and one just woke up and the other guy just had some water, maybe that would lead, that could lead to these differences in percepts. So we're hoping that if there is a system, this will give us the ability to clamp it and maybe reduce some of the noise in the brain. Okay. So because of that problem of, the system, how do we find the interesting computations? How do we find internal state computations? Because I know that there's more that are going on, not just here in the nose. The whole thing isn't just about the nose. The brain is important, right? And what we believe is that one of the things that may be slowing us down in the social behavior field is that we have a history of sort of knowing where the action is in the amygdala and the hypothalamus, throughout the limbic system, and then these motor command regions. And right now we have different groups that are sort of focused on them independently. And what we'd like to do, instead of just focusing on a particular node, we think that maybe, if you may find some internal state modulation on a particular node, but you still don't know what's happening in the rest of the brain, right? So if you're just focusing on one region, you may see it as a wall or a tree or a snake, but it's really an elephant, right? That's sort of the thought here. And so for the last few years, we've been sort of obsessed with trying to get we don't have a zebrafish where we can look at the whole brain simultaneously. And so what we wanted to do was get sort of a full parts list of a behavior. Is there such a thing as a primary sort of a skeletal circuit where everything else sort of feeds into it? Is there a primary circuit? Is it definable? Can we make a circuit diagram of it? And can we find the complete circuit from sensory to motor for a social behavior? And the thing that really stymied for this, as a state of thought experiment for a really long time, was that most of these social behaviors are complex at the sensory level, so olfactory cues feed into them, but also other things, as we've heard, somatosensation and auditory information, and even sometimes visual cues, right? And then at the output level, as you saw, Dayu has this great jaw motor action, but aggression is composed of a lot of other motor behaviors, and they're all dynamic, and all of these behaviors can use up to tens or hundreds of different muscles in their motor output in a dynamic and unpredictable way. And so both at the entry point and at the exit point, finding a complete circuit was really daunting to us. And so that was until a graduate student, Jason Keller, joined my lab and convinced me that we should be looking at this very simplified behavior. Okay, so this is a social behavior that's occurring with just one animal. And so what we did was spot the smell of a female here. And when we did, he got a whiff of it and he started marking. So the female odor here has her pheromones. And he is super excited. He wants to have her come and hang out with him. And so he is advertising to her by marking with his own urine cues that are now wafting out pheromones and hopefully will attract this female. Okay, so this is the behavior that Jason thought would be the model circuit that we could solve. And I really didn't want to be the lab that was focused on urination. just because it's not as glamorous or exciting or as interesting or apparently as complex as all of those other wonderful social behaviors that you've heard about earlier this morning. But I'm going to tell you why this was, I think, a really good choice. Okay, so the first is that there's two different kinds of urination. So when you're young, you do reflexive urination. That's based on physiological need. Your bladder gets to a certain pressure, and you just eliminate. And then once you've developed, both mentally and physically, the rest of our lives, hopefully, we get to do voluntary urination. And the definition of this is that you go when you don't have to go, and you don't go when you do have to go. So if you can control it under those two axes, it's voluntary. What I'm going to propose to you is that that's what these mice were doing. So in that video that I showed you, that was a compilation of a 4-minute assay. So we do 2 minutes with the control odors, and these are 12 different mice. There's little rasters whenever they've urinated. And you can see that after we drop in the female odor cue, there's a high probability of them urinating. So they didn't have to go beforehand, and they all suddenly went, and so we would call that voluntary urination. The other thing about this is, just like those other behaviors, this behavior is modified in a lot of different axes. So it's modified by development. So juveniles don't respond to the female odor cue with countermarking at all, with marking at all. They don't do the behavior. It's gender dimorphic, so females won't mark in response to female urine. It's based on experience, so subordinate males respond differently than dominant males.
I'm struggling with voluntarily as opposed to triggered by a certain cue. If a behavior is triggered by a certain cue, would you still call it voluntary?
It's not based on, it's not a reflex, right? So all of these cues promote behavior. All of these pheromone cues promote behavior with a very high probability. So you could say that aggression would be reflexive in response to these cues, right? But what I'm saying here is that it's not, so that reflexive urination that you do until you're 2 or 3 is all in the spinal cord. And this, I'm saying, is going to use the brain.
Okay. No, but I'm not worried about reflex or not. I'm just worried about voluntary. If you say a behavior is cued by something else, not reflexive, but just cued, meaning you release it, the behavior is released if there's another certain trigger cue.
Yeah.
This doesn't have to be a reflex, it just can be a cue that releases the behavior. Is that what you mean by voluntary? Or
actually, no. So,
yeah. Voluntary has this notion of there's something inside that makes me choose or not to choose, so it sort of has a slightly a flavor, a psychological flavor that from a purely mechanistic reductionist standpoint, I'm struggling with.
No, all of these words can be distasteful, right? But in this system, in the field of continence and incontinence, voluntary urination has this meaning that you urinate when your bladder is below a certain pressure. So when you don't have to go, when there isn't a pain, and that's what's occurring here. Mm-hmm. They're not just. This is in response to the stimulus. And the stimulus is making them want to go when they don't have to go. And that's just like, your stimulus is going to be the restroom. You see the restroom and you decide whether you're going to go or not. Your stimulus is, the restroom sign is like this odor cue. And the males choose to go in response to this odor cue, but juveniles don't, and females don't. And subordinates do it differently. That's all that I mean.
Following up on that question, I would say that it will be interesting if you have mice living in their social group together, so you have male and female together. I would say that the male will not urinate when he smells the female odor, the odor of the female in his group, but he will urinate when he smells a female from outside of his group. That's going to be like a criteria to test voluntary urination. So
yeah.
he get the same female odor, but he recognizes different individuals and decide when he wants to urinate or not.
So that would be a fun experiment to do. But if we do it now without knowing where in the brain to look for, right? Without having our circuit, all we would be able to do is say, this is the result that occurs behaviorally, right? And what you really want to know is, Why would the male not respond to, what is the mechanism in his brain that makes him not react to the females that he knows versus the females that he doesn't know, if that's the outcome? And so that's the kind of second order question that we'd like to ask once we have a complete circuit, but to ask it now wouldn't get us anywhere. But these are the types of things that you put on the list and say, that computation is in there someplace. It's not just a reflex.
Yeah, exactly.
Yep.
And if you're interested in intentional communication, that's going to be a really interesting thing to test.
I agree. Okay. And then this behavior is also modulated by different sorts of internal states or homeostatic states. So if the animal's thirsty, if it's sleepy, if it's stressed, it all alters this pattern. And so these sorts of decision-making or computations or features of the circuit are rich enough to us to make this be a nice model behavior to be able to invest in. And then finally, unlike the more fabulous social behaviors, this behavior is gated really simply on a single muscle unit. Okay, so when you urinate, what happens is your bladder contracts, and there's a urinary sphincter, which is this awesome muscle that basically contracts for most of your life. It is contracting now in all of you. And only in those few times when you urinate does it relax, okay? So the key to this is that the bladder contraction the bladder is composed of smooth muscle, and that is autonomic. So it's not voluntary. So if you want to urinate right now, you cannot squeeze your bladder to do it. You can squeeze your abdomen, but you can't use your brain and squeeze your bladder. But you can, what you do is you're relaxing your sphincter. This is striated muscle, it's voluntary, and this is what your brain is doing, right? So all we needed to do was find the neurons that control the single muscle, and then our output is simplified. Our input is simplified because we have this female odor cue that's sufficient. So now we're through one sensory system, and so all of these things together made this really nice behavior. And when we first started it, I was also super excited because I'm in a cohort of people whose parents are getting older and whose, everybody's had kids, and there's been a lot of talk of incontinence in our life, and it turns out that incontinence is this horrible medical problem. So 30% of Americans become incontinent in their lifetime, so that pretty much means that either you or somebody you know or somebody in your family is going to be impacted by it. So I thought that there would be a wealth of circuit information. All we would have to do is plug the mouse olfactory system into it. We'd have our circuit and we'd be decoding, and it turns out that wasn't the case. So when we first started this, as you guys all know, the brain is important for voluntary urination, using it in the urinary sense here, not in the neuronal sense, voluntary urination. And so if you suffer an accident, have spinal cord injury, you're reduced to reflexive urination. And so almost all of the literature on the mechanisms and the control of urination is studied from the spinal cord down. And what we knew at the start of this was that there was a region in the brainstem called Barrington's nucleus that had been shown in several animal species to be important. But Barrington's nucleus is heterogeneous, and even the cell type within it hadn't been described. And so basically we were faced with having to find this whole circuit de novo. And so what was known was that this subset of cells in red, they express the marker CRH, and they were thought to be the primary control of urination within this nucleus. And we had tested these, the response of these neurons during our odor-mediated behavioral paradigm and found that they didn't affect the system much. And so we went ahead, used a candidate neuron approach, and found a second population of neurons in this nucleus that just happened, with the theme of the day, to express estrogen receptor. But we're going to use that just as a marker here. And so these green neurons, these estrogen-expressing neurons, are largely distinct from these CRH neurons. And if we trace from either the green or the pink neurons down into the brainstem, we find two different patterns. And so this pattern was previously described by Bernardo Sabatini's lab from these pink neurons, and it ends up labeling neurons in these two focal regions here in the medial lateral region of the lumbosacrospinal cord, and we recapitulated what he found. And this region is known to innervate the bladder. And if we trace from the green neurons, you can't see it here, but it's also showing these two focal regions, as well as this region across this dorsal gray commissure, and that's known to control the sphincter. So these two different populations of neurons have different molecular markers. Bernardo Sabatini had already shown that these neurons do affect the ability of the bladder to contract, and we were wondering if these were also involved in urination. So we did some fibrophotometry with these neurons. This is a trace of the change in fluorescence from those small population of those green neurons, those estrogen receptor cells, and this is the behavioral response of urination events, and you can see that we get a pretty nice correlation between the neural activity and the behavior, as shown here. And so if we went ahead and did activation of these neurons, and this time, so this is sort of the same type of situation that I showed you earlier, but we have a camera from below. So that was just a very low frequency stimulation. So if we go ahead and do it at 5 hertz, you can see that from below, you can see there was immediately a release of urine. And if we go ahead, let this animal move around again, and do this in a frequency dependent manner, we can find that these cells are indeed promoting urination. So the pattern that, the place that they were in the brain and their localization in the spinal cord is consistent with the role in urination here. So there is these two different population of neurons. So this is sort of the data that I just showed you here. So these two different population of neurons, and if we do the same experiment with those other cells, those CRH expressing cells, we can see that occasionally we do get some urination events, but it's a little lagging from when we turn on the light, and the amount of urine that we get is very few and very few neurons. So this is just from the neurons that we do see urination from, whereas we see a really robust response from these ones that are also these green neurons. From this brainstem region, we can also get this sort of urination behavior in anesthetized animals. And so if we just use channel in those green neurons here, so the light is on and you can see that urine is coming out. And at the same time that we're stimulating with light in those green neurons in the brain, we can also record the pressure in the bladder and do an EMG in the sphincter. You can see the bladder pressure rises and the muscle starts pulsing. So it's getting a very strong contraction and then relaxation. And that pulsing is coinciding with the release of the urine. And what's actually happening is that the urine's being released during these periods in between the pulses, so that it's like the urethra is really small in the mouse, in the male mouse, very narrow, and it's like a peristaltic pump is what's happening there. And so basically, these two different populations of neurons, you have these pink population of neurons. If we do the same anesthetized recording of bladder pressure and sphincter activity, when we stimulate those pink neurons, we see the bladder pressure rise really nicely. It rises whether we do it from those pink neurons or the green neurons. And in fact, we see a large rise in bladder pressure from those pink neurons. And then it decays after we turn off the light back to the initial starting pressure. And that's just like taking a closed balloon and squeezing it, where the pressure is going to get stronger, but that's just because of the surrounding pressure that you're doing with the squeezing. That's happening with the bladder pressure, but it's not productive. And here you have the same rise, so the same effect on the bladder from these green neurons, but it never gets high, and that's because the urine is leaking out. And so at the end of the lights off, you actually see a decrease in bladder pressure, and that's because it was productive. Okay, so we found cells that potentially have the capability of being mediated, and this odor-mediated behavior, and the question is, were they? And so in response to odor, we first just dreaded the system. So with those green neurons, you can see on days when we injected CNO, we basically got no urination whatsoever in response to the female cues, and on days when we injected saline, we got nice urination events. And so that suggests that the odor-mediated information is using these green neurons. We could go ahead and instead of inhibiting by DREADS, we can use light-mediated inhibition. So Jason starts with lights on. There's the control odor. And then this guy can venture out. And you can see that it looks like he can still sense the cue, and he's interested in it. But he doesn't do that same robust marking behavior that I showed you in the other videos. So there's a little bit that leaked out there and a little bit that leaked out there. And then he turns the lights off. And I don't have the camera from underneath, but you can already see the glow. He starts marking right away. And then you can see he did a little sniff there, and he starts resuming the marking right away.
And by the time that the pee was shipping,
Yeah. So pooping is interesting.
Is there, but couldn't
Yeah, so yeah, pooping is very interesting, and so I mean, pooping is controlled by this nuclei as well, and sort of a lot of different pelvic floor behaviors. We don't see, quantitatively, when we've looked at pooping, come join my lab, it's really awesome. We don't see a correlation between neuronal activity or inhibition and pooping, but we do, you know, in our anesthetized prep, we do sometimes see rhythmic anal activity. Yeah, so we think that there may be something going on there, but we have not studied it, and we don't, these cells are not sufficient for that. Okay. Okay, so this is just what I showed you, and what I want to reiterate is that that video wasn't an anomaly. Okay, so this is when the odor cue went in after lights on here, and all these blue marks are when the animals sniff, so all the animals still seem motivated to be able to sniff, so that seemes to be controlled upstream of this nuclei, and then that ability to, that bout of urination seems to be, in many cases, occurs right when the lights go off, that there's a strong response. You know, that could just be a technical problem with some sort of a rebound activity, but if we go ahead and do lights on, lights on, lights on, lights on, lights on, lights on, without the odor cue, we don't get that rebound activity. There's a little peeing here after this long event. And so we really think that the system is sort of the sensory cues are in there. There's something else that's sort of kindling within the upstream circuits, and that it really needs these nuclei to be able to progress. Okay, so what I've shown you is that we found this anchor point to be able to find this model circuit. These neurons are nice, they're linked to our odor-mediated behavior. Surprisingly, we found two sets of neurons that are controlling the bladders, one that's doing it without being able to, it just squeezes the bladder. We think that this is a really odd function of these cells because bladder pressure is very uncomfortable, and so having the cell type that's what it does, we don't see a purpose for it. And so we're curious why there would be these two populations of cells.
But they have different targets,
no? Both of them are going to the bladder, and they both squeeze the bladder. And then one of them, the green population, also goes to the sphincter and simultaneously relaxes the sphincter. So we can imagine that there are they're going through the spinal cord, and there's a lot of ways where the spinal cord could be.
Do you know if it's the same neurons, the same green neurons that both go to the bladder and the sphincter?
So there's a relay in between in the spinal cord. So the green neurons are going to the right. So no, I think there's slightly different subsets, but we don't know for sure. We have not been able to separate.
It could be that it's a subset of green neurons that only go to the sphincter and another subset that only goes to the bladder, or not?
It totally could. This is a very small population of cells, and so molecularly we would like to know that, whether they're distinct, and we don't know that, but there's only about 200 of them total. So we could subdivide it even more and perhaps find just the sphincter neurons.
Or just the bladder neurons.
Right, but the sphincter neurons are the ones that we want for this behavior.
But it could be that the green neurons, that you only have this nonsensical subset that only goes to the bladder but doesn't go to the sphincter. You said it makes no sense, right?
That's the pink neuron. Only the pink neurons are the ones that don't make sense to us.
No, but I'm asking whether also amongst the green neurons
there could be some nonsensical
exactly
ones
the
as
same
well.
ones that just go to the bladder and not to the sphincter.
Yeah.
And I think one way to make sense of it is that you have neurons that just have a specific target. That they can only have one target because that's their job to go there, and that you have another subset that you can't distinguish yet that only go to the sphincter, and that's the way they've been wired up, and how you control it, you just have to coordinate them, right?
Yeah. So we think that yeah.
Sorry, so I would also love to discuss maybe with you afterwards, but I'm just wondering, because in the male mouse, so there are different pelvic muscles as well, yeah, which are important for erection, ejaculation. So my first question would be if you see spontaneous reactions of the male mouse due to this female pheromone. And my second question is if you are, so there are many questions. Anyway, so if you ever try to label the motor neurons from the sphincter to the spinal cord, and also if you think that the sphincter is the only muscle controlling the urination, because what we study is actually this bulbospondiosus muscle, which is supposed to be the main muscle for ejaculation, but I'm not so confident anymore that it's actually only for ejaculation, and it might play a role also in like holding the urine or like expelling the very last rest.
Yeah, so this nuclei has been implicated. So it's got another name, which is the Pontine Micturition Center. Micturition means urination, and we use its classical name, which is Barrington's, because it does more than just urination. And so it's also been implicated in also all these other pelvic floor muscle behavior or responses that the animal does. So defecation, as we mentioned before, during childbirth, you have to control the pelvic floor muscles, the sexual activity. And so what we can imagine is that these two sets of neurons, while these green neurons for us with this voluntary behavior are the primary focus for what we're interested in going further, this conundrum of why there's these two populations we think can be resolved by these other activities and the coordination. They all coordinate. During ejaculation, you need to close the sphincter so that you don't have ejaculate reflex. During childbirth, there's other considerations, and that all of these behaviors are being coordinated through these subsets of neurons, and we haven't studied that. We have been able to see ejaculation a couple times when we stimulate this area in the anesthetized animal, but nothing reproducible.
It seems that, I don't know, it seems that the ability to increase blood pressure without opening up the sphincter is important during normal male aging. Right, so if you're a 75-year-old man, typically you can't pee because you're jumping on stage, the reds are from getting out. The way you deal with it is by squeezing your bladder without opening the sphincter, dramatically increasing blood pressure, and then voluntarily loosening the sphincter when this dash of urine comes out, and you stand in the bathroom for an hour. So your urologist goes in there and puts it out. Actually, my dad actually has a meter that.
Right, so I didn't know about that.
I'm actually wondering whether or not aged male mice are getting large prostates. Actually, mouse is
so we have a colleague that's looking at a model of something like that. Now that you've described it, I understand it better. And it could be that there's something like that going on, but there are multiple things that come out of this. And so we had a really high probability behavior. When we stimulate, we see urination pretty much every time. So you go, oh, well, at this part, we're in the brainstem, these neurons, you now have a labeled line to urination. And we think even at this point, based on this discussion that we've just had and what we know about this, that even at this point, we don't think it's a labeled line. We think that these neurons can be doing a lot of different things and used in a lot of different contexts. Yeah.
I mean, what Bob just said, you might also want to control pressure independently if you want to regulate flow. Like if you wanted to release, or just, I don't know whether for male mice it also matters how far they can pee, if they mark stuff, whether they need to have large trajectories or shorter ones. But I'm sure for some animals, for dogs even, if you watch this makes sense, right? I think that might be another way how having control over the pressure voluntarily or by cue dependent might make sense, no?
Yeah, no. I mean, we can imagine a lot of different and we can imagine other modulations that feed into the system. You know, these cells are expressing CRH, and that's been implicated in the system in different ways. And so we can imagine that there are different instances when these cells could be used in these neurons would be used independently, or how using them judiciously and with different timings could alter the behavior. For us, from the perspective of trying to find how we link productive olfactory mediated behavior to this small subset of green neurons is good for me at this point. And because we have this nice marker, and it's only 200 neurons, we've been able to trace backwards from it, and we've been able to sort of flesh out a nice skeletal circuit so they don't project all over the brain. They're not getting feedback from, you know, the whole brain. They're getting feedback from very few areas. So this is where we were, the spinal cord and the muscle. And as we work backwards, we find a very few regions. Some of them we have questions about. We're not, whether they're all equally important in this behavior, we're still working out. We know the nose is involved, and these gray boxes here are hypothetical. From here down, we sort of have it worked out. From here backwards, we have it worked out. This is the shortest path that we are testing right now. And so if this is the case, then I would suggest that we could have this primary circuit, and then we could start to study it for interesting things. And I think I'm out of time, so I'm going to actually skip this whole next section, which was really cool, on ultrasonic vocalizations, and just get to this last thing. Okay, so and what I'm going to tell you is that we ran into another circuit. While these mice are marking with their chemical cues to attract the females, they're also singing, doing these ultrasonic vocalizations to attract the females. So they're using two different modes of communication. They're both triggered by those olfactory cues. And so we ran into neurons in this, here it's called it's really the lateral MPOA that is going into these calling centers. Ultrasonic vocalizations are fairly well understood from the PAG down. This is sort of a new node that's upstream in the hypothalamus that we found. And from there, the pathway is really well worked out, so we're able to just plug into that. So we now have two different primary circuits that are elicited by a simple sensory cue. They impinge on single muscles, and they're modulated. Calling is modulated by some of the same characteristics and features that I told you about with the urination. And so we can use these systems both simultaneously to look for different types of computation. So we don't even know the flow of information. Is it primary feed forward? Is it dynamic? We can be able to mark the motor events, and so I showed you that they were sniffing, they were walking. I didn't show you that they were calling, but they're calling and they're peeing. We can take this maybe into a little more detail, but they don't do too much else as far as motor events go. And then the types of things that interest us are these features. Whether these words are good or not, I don't know, but these animals, they have a different sort of arousal level after they've interacted with the stimulus. We'd like to say that it's somewhat positively valenced. They're quite relaxed and happy. There's these sorts of different words that you could use to characterize some of the features of behaviors. And then some things that may be easier than these sorts of non-mechanistic words are sort of these things that modulate in ways that we could imagine would have an easier mechanism, and these are in both systems too. And so we'd like to be able to map these and ultimately understand the coding of them. I don't have any answers at this point. But by having the two different systems, we can imagine that if we have a mechanism for how stress silences the system in one, we can look in the other. If we understand it in the urination circuit, we can look at it in the calling circuit and ask, are they using a similar mechanism and a similar property of the neurons, or have they evolved in two entirely different ways to interpret the response to stress? Right? Okay, so these are the guys that did it. These are guys that we collaborated with, and these are the guys that gave us money. So thank you.